Hi, hello everybody. Okay, so before the March holidays, we spoke about the idea of carbohydrates, and carbohydrates actually having three types of carbohydrates. Number one will be your single sugars, that means they only have one sugar, okay, and the common one is actually glucose. Next, we talk about double sugar. That's why the term double, because they are actually made up of two sugars and there is a chemical bond that is in between them, okay, and the most common of them all is maltose. All right, then we talked about complex sugar. Now the word complex sugar actually just means that there are many, all right, single sugars that are chemically bonded together. You can see all these small little lines here that Miss Lee is pointing your attention to. Now these are chemical bonds that attach the sugars together. Now there are a few types of complex carbohydrates that you will need to know, and they are your starch as well as your glycogen as well as your cellulose. Now, before the school holidays, we actually spoke about the difference between all three of them. We say that starch is basically found in plants and it is a storage form of carbohydrates. That means plants keep extra carbohydrates in the form of starch. Cellulose is also found in plants, and because of the word cellulose, it sounds like cell wall. Okay, so your cellulose is actually found in your cell wall. In fact, it makes your cell wall. The last one is actually glycogen, and glycogen is found in your animals, and it is a storage form of your animals in animals. We also mentioned that all single, okay, and double sugars are reducing sugars except sucrose. That means sucrose is not a reducing sugar. Okay. All right. Next thing, we talked about where your single and your double sugars can be found. All right. We also talked about how your double sugars can be formed. Look at this. Two, okay, separate single sugars known as glucose. If you want to form a maltose molecule, there must be a chemical bond in between them, and then after the chemical bond is formed, it will be called your maltose. Now your maltose is actually made up of two glucose, okay, two glucose that are chemically, all right, bonded together. All right. Next thing, if I have a maltose molecule and I want to break this down to give me two separate sugars, all right, what I will probably need, okay, is water and a scissors. Now this scissors is known as an enzyme called maltase. So what maltase will do is that maltase will cut this chemical bond. We use the word cut, but actually in biology we say we use the word digest. So it will digest the maltose to give me two separate glucose molecules. Okay, then of course we spoke about the three different complex carbohydrates. Now we're going to move on to the next part, which we ended our previous lesson. We spoke about what fats are actually made up of. Now fats are organic compounds. Now why are they called organic compounds? Because they contain your hydrogen molecule. Now they are also made up of the same three elements that are also found in carbohydrates. Okay, they are also made up of the same elements that are found in carbohydrates. The only difference is that fat contains much lesser oxygen in proportion. That means every fat molecule will have lesser oxygen than one molecule of carbohydrate. Okay, now if you look at this animation here, it basically shows you what fat molecule is made up of. You can see that your fat molecule is made up of one glycerol molecule, and it's made up of 1, 2, 3. Now these are known as your fatty acid chains. Now why do we consider them as chains? Because they are very, very long, just like a long chain or a necklace chain. So this is one fat molecule. So inside your cooking oil, you have many droplets of fat, right? So every single droplet of fat is made up of many fat molecule. Okay, Now, we said that what is the function of fat? Fat is a very good store of energy. What's the meaning of store? Means that it allows energy to be kept. So if your body wants to release energy, it will basically break down, okay? It will break down fat and will release, okay, and will release energy. It is also a source because it allows your fat to be broken down. The next thing is that fat is a very good insulator. Insulator means it will prevent, okay, heat from being lost to the environment. That is why your penguins, okay, or your whales, they have a very thick layer of fat because they stay in very cold countries. Alright, next thing we say that fat is also a solvent for fat soluble vitamins. Okay, it is also a solvent for fat soluble vitamins and some hormones. That means these vitamins and these hormones will actually dissolve, okay, it will actually dissolve in your, alright, in fat. Next thing, it also forms the main part of your cell membranes. So all your partially permeable cell membranes will basically contain a large proportion of fat. It will help to reduce water loss. Now, how does fat actually help to reduce water loss? Take, for example, this is the surface of your skin. Now, this is a layer of fat here. Now, can you remember when you were in secondary 1 or 2 or in primary school, if this is a beaker of water, your teacher will always say to prevent the water from evaporating, you must put a layer of oil. Now, that is because, okay, water cannot mix with fat. It is immiscible. So because this is your skin, alright, and this is a layer of fat, what your fat will do is that it will prevent, okay, it will prevent any water inside the skin from exiting. So water cannot come out of the skin. That's why it helps to reduce water loss. That's why, you know, girls, we like to put moisturizer, right? Why do we say we put moisturizer to prevent dry skin? Because dry skin is caused when too much water is actually being lost from the surface of the skin. Now, wow, look at this yummy picture here. Are you able to list some food that is very high in fats? Maybe you can take some time to write this down in your notes. Okay, I hope that you have actually listed some food or be able to identify some of this. This is very good. Butter. This will be known as cheese. This will be your favorite peanuts or your almond nuts. This is all our favorite we eat in Korean barbecue. Okay, for some of your friends who can eat pork. If not, this is actually known as meat. Now, pork, okay, or even beef, or even fish will also have a lot of fats. Alright? Now, just like how we decided to break down your carbohydrates, how do we also break down your fat molecule? Now, you see again, this is your fat molecule. We have one glycerol molecule, and we have how many fatty acid chains? Yes, you are right. We have 3 fatty acid chains. So because we have three fatty acid chains, we also need 3 molecules of water. Now you see this enzyme again. Remember Miss Lee said this enzyme is a pair of scissors. So what these scissors will do is that it will break all the chemical bonds. And because it breaks all the chemical bonds, now all these are basically free. Okay, they are already detached from the glycerol molecule. Alright, now this is known as hydrolysis. Now I break down the word for you. Hydro, because we want to use water, because water contains hydrogen molecule. Then lysis actually means breakdown. Okay, it means breakdown. So you are using water to break fats. And that's why you get 1 molecule of glycerol, okay, and 1, 2, three fatty acid molecules. So if Miss Lee were to ask you, what is the building, okay, what is the building block of fat molecule? It will be a glycerol molecule, as well as 1, 2, 3 fatty acid molecules. So 1 plus 3 will give you a fat molecule. Okay, sorry, this is a wrong slide, shouldn't be here. All right, now that we have finished, okay, the chapter of carbohydrates and fats, we are now going to talk about proteins. Okay, so can you please turn to 4.5 of your notes to look at what are the functions or what are the elements that make up protein. Okay, so at the end of this section, you must tell me what are some of the elements of proteins. Now, we've discussed what are the elements that make up your carbohydrates, right? C stands for carbon. Yes, you are right. OH stands for hydrogen. And O stands for oxygen. Good. And then we also just spoke about fats, right? And we say that fats also have the same elements in carbohydrates. C stands for, you are right, carbon. H stands for hydrogen. And O stands for oxygen. Good. But what is the difference between the O in the fats and the O in the carbon? Okay, carbohydrates is that the O is actually lesser in proportion. That means one fat molecule will have very little oxygen. One carbohydrate molecule will have a lot of oxygen molecules. So now we're going to be talking about the third nutrient known as protein. Then we are also going to be talking about the building blocks of proteins. Then we are going to find out how to detect proteins and also how to detect carbohydrates and fats. Alright, so just now we spoke about the hydrolysis of fats, right? Now we're going to be talking about proteins. So what are proteins? Now you watch, Do you notice anything that is similar? Very good. If you told me that you notice, okay, that carbon, hydrogen, and oxygen are also present in carbohydrates as well as fats, you are right. But do you also notice something that is very different? You will also notice that protein has got two more additional elements. These two elements are new. And what are they? They are nitrogen. Nitrogen, the chemical element is N. And they are sulfur, known as S. That means protein is actually made up of carbon, hydrogen, oxygen, as well as nitrogen and sulfur. Wow. So these are actually new. These are similar inside your carbohydrates and inside your fats. That means your fats, okay, your proteins, your fats, and your carbohydrates all share three same elements: carbon, hydrogen, and oxygen. But nitrogen and sulfur are only found in protein. Okay, carbon and carbohydrates and fats do not contain nitrogen and sulfur. Now, protein molecules are made up of basic units known as amino acids. So earlier on, we say that your carbohydrates are made up of glucose. Okay, your fats are made up of fatty acid and glycerol. Okay, now we say that your proteins are made up of amino acid. So your amino acid is like the brick that makes many proteins. Okay, so what is the function of proteins? This is something that you will need to know. So Miss Lee is going to highlight some of the important key points for you. So what is the function of your proteins? Okay, your proteins are needed in the synthesis. Now, synthesis means that you want to make something. So what are your proteins going to be made? What going to be used for? They are used to make new protoplasm for growth and repair of worn out cells. That means these are your cells that need to be replaced, so your proteins need to make new cells. Next thing, your proteins are also used to make enzymes and some hormones. So they are very important because your enzymes are involved in the process of digestion. Next thing, your proteins are also used to make antibodies. Now, what are antibodies, you may ask? Antibodies help to fight your basically they make up your immune system. They help to fight off bacteria. Now, there's this whole thing about the COVID virus, right? So the COVID virus actually requires a lot of antibodies in your body to fight it off. All right. Are you able to list some food that are high in proteins? Maybe you can take 2 minutes to decide what are some of the food that you are eating every day in the canteen or even at home or even now. All right, that is very high in. All right, so what are some of the food that is high in proteins? All right, your fish, your salmon fish, okay, your beef, this is your tofu, okay, your soya bean, your bean curd, as well as eggs. Now your eggs don't look like this, but okay, your hard-boiled eggs are very important source of protein, and that is why you know all your bodybuilders, they want to actually build more muscles, they will eat a lot of protein. So I'm going to skip this portion. So let's now talk about what are proteins. Now on the picture over here, you can see, right? You can see that there are many different shapes and there are many different colors. Okay, so what do the colors represent? All right, now the colors actually represent amino acids. Okay, the shape represents amino acid. Now you notice that there are different shapes. You got what is this? A rhombus shape. You have an oval shape. You got rectangle and triangle. You got 4 different shapes here. You also realize that there are 4 different colors. So what does the orange rhombus, the blue oval, the green triangle, and the purple rectangle mean? It means that you have different. Now this is very important. Okay, there are many different amino acids in your body. So these different amino acids will then be chemically combined. Can you see this line here? Okay, this line actually represents chemical bonds. So all your amino acids will be chemically bonded, okay, to give you your protein. So your amino acid is what we call the building block. So why do we use the word building? Block is like your bricks. Why do we say building? Because you need your amino acid to build one by one, like a brick like that. You see what Miss Lee is drawing? Okay, like a brick to actually make a house. So you need your amino acid to be put together in order for you to get a protein molecule. So once you get this long line, okay, this long line of different shapes and different colors representing different amino acid, we say that this thing is known as a polypeptide. Because you can see that it is still like a straight line, right? It's not bundled up together. So all these different bonds here, okay, sorry, Misty is going to erase it to make it clearer for you to see. Okay, maybe it's clearer now. All right. So you will notice that you have many chemical bonds, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Now, what are all these chemical bonds? What are the names? They are known as peptide. Why do we call them peptide? Because it starts with P, just like proteins. Okay, so when we hear the word peptide, we know that it's got something to do with protein because P and P, protein. So all these peptide bonds will join all the different amino acids, okay, together to form a polypeptide. Now, why do we use the word poly? Poly actually means many. Now, you know Singapore got many polyclinics, right? Why do we say polyclinics? Because there are many clinics in Singapore. So why do we say polypeptides? Because there are many peptide bonds that are joined together. So when all your amino acids are joined together, we say that it is a polypeptide. Okay? You can look at this molecule over here when there's more than one polypeptide chain. So if you have 1, you have 2, if you have 3, So 1 polypeptide chain, 2 polypeptide chain, 3 polypeptide chain. If you join them all together, then you will get a protein molecule. And the protein molecule is a 3D shape, so it looks like a ball. You can see over here, it looks like a ball of wire, right? But over here, it still looks a little bit neater, like you are queuing up in a straight line. But over here, you can see that the straight line is all jumbled up together. So when it's jumbled up together, then you get a protein molecule. If it's still a straight line like this, but still got a lot of chemical bonds here, we say that it is a polypeptide chain. Okay, so I give you some time to copy your notes first. You may want to pause the video here. All right, so now let's move on. Now at this point, okay, we are now going to ask ourselves, now we've spoken about carbohydrates, we spoke about fats, we also spoke about proteins. Now the more important thing is, how do we identify for the presence of these nutrients in our food? Now in Singapore, we have something known as the AVA. Now the AVA is basically a place in the government that identifies different types of nutrients in our food. That is why in your food, right, your nutrient label, you can see that they are able to identify sugars, fats, proteins, certain types of minerals, and certain types of vitamins. Now, why are they able to do that? The answer is pretty simple, because they are able to run tests in the laboratory to identify for the presence of these nutrients. So today, we are going to be looking at some of the tests that are required to actually identify for this. Okay, now Miss Lee is going to show you a few videos. I will probably only be able to show you 2 videos because we will then need to complete your notes. After this, you will then have to proceed to your SLS platform, okay, to complete a couple of lesson packages. All right, so I'm going to pause the video here, and then I'm going to start the video again to show you a couple of videos. All right, so please pay attention. Okay, so Miss Lee is back. Now, I'm going to just show you 2 different tests that we'll be carrying out in the lab. So basically, in your practical, and this will also be tested in your MCQ, you would have to look at a few different types of tests to identify reducing sugars. Now, if you look at your notes, reducing sugar is actually your carbohydrates. So there are 2 different types of tests to identify for your carbohydrates, okay? Now, the first test is actually known as your reducing sugar test. So please have a look at this video. Okay, this is a test for reducing sugar. So we will need to use Benedict solution for this test, okay? To a clean test tube, add 2 cm³ of the sample, in this case, glucose solution. Add an equal volume of Benedict's solution to the sample in the test tube. Shake the test tube well. Place this test tube into a warm water bath and leave for 2 to 3 minutes. So if reducing sugar is present, you will get a change in color. Okay, Benedict solution will change from blue to brick red or any other color. If the sample does not contain reducing sugar, Benedict solution will remain blue. So over here, all right, you can see a picture here. Basically, the picture shares with you that there are different colors, right? So this is a blue solution. That means this is a negative test. That means there is no reducing sugar. There's no carbohydrate here in that sense, right? Because remember just now, if you look at your notes, Miss Lee mentioned that reducing sugars are usually your single and your double sugar except sucrose. So over here, you won't see any reducing sugar. But if the blue solution changes to green or changes to orange or changes to brick red, it basically tells you that the reducing sugar is present. And why are they different colors? Because they're getting more and more concentration. That means the reducing sugar is becoming more and more. If it's green, that means traces. Traces means only very little bit of reducing sugar. Orange means it is very moderate. That means it's a higher than little bit of reducing sugar. If it's brick red, it means that there is a lot of reducing sugar present. Okay? So back to your notes, because we must be able to know how to carry out the experiment. So we carry out a Benedict's test to test for reducing sugar. Okay, now the type of reducing sugar that we usually test is basically glucose. All right, so we always test for glucose, and you know that glucose is a type of carbohydrate. So we will add 2 cm cube of the Benedict solution to 2 cm cube of the food sample. Now, usually we will use a sample because actually in the lab, we might have an unknown solution. So we will use an unknown solution, but you notice that the volume of the Benedict solution and the volume of the sample is exactly the same. Then we must shake the mixture very well. We will then put the mixture inside a boiling water bath for 2 to 3 minutes. Okay, that was what you actually saw, right? This is a picture. Now, if there is a large amount of reducing sugar, you get a brick red precipitate. So if an MCQ question tells you that you use a solution X, and then in the solution X, you put Benedict's solution, carry out the Benedict's test, then they say that, oh, the test will eventually become brick red. What does that tell you? It tells you that reducing sugar is present. If, okay, the Benedict solution remains blue, that means reducing sugar is not present at all. This was the picture that you saw just now, right? So this means it is a negative test. Reducing sugar is not present, it is absent. But if you have green or yellow or orange or red, it basically tells you that reducing sugar is present in different amounts and different concentration. That's why I got different color, because different color tell you different things. Okay, so remember, blue solution means no reducing sugar present. Any other colors after that tells you that reducing sugar is present, but the amount of reducing sugar is different. Brick red means the highest amount of reducing sugar is present in your brick red PPT. Alright, so what does PPT stand for? It means precipitate. Okay, now we are going to stop here. Tomorrow, we are going to continue with the different tests to find out all the different other nutrients. So today, we actually covered quite a bit of things, right? We covered basically, okay, we basically covered what are fats, what are the elements that make up fats. We also covered what a fat molecule is made up of, right? You can see it's made up of your 1 glycerol molecule and 3 fatty acid chains. We also covered what are the functions of fats. We also covered the types of food that contains a lot of fat, your butter, your cheese, your meat, your nuts. We also covered how to break down fat. So we need to use an enzyme. The name of the enzyme, we will talk about it in your chapter of digestion. We also mentioned what are proteins, and we said that your proteins are made up of the 3 same elements that make up your carbohydrates and fats, okay, your carbon, hydrogen, and oxygen. But the new ones only inside protein are nitrogen and sulfur. We also said that your amino acid is the building block, right? It is the building block of proteins, and we covered what are the functions of your proteins. Basically, your proteins are used to make new cells because your cells are either spoiled or they need to be made new. Your enzymes and antibodies are also used to make muscles, by the way. Okay, we also mentioned that your amino acids, different amino acids will join together by peptide bonds to form polypeptide. Your many polypeptides will come together to form a protein molecule. We also talked about the different tests, especially your Benedict's test to identify reducing sugars. Okay, tomorrow we will continue with the rest of the lesson. So now please proceed to SLS to complete the other part of the lesson. Thank you.